Okay, it works. So, yeah, this looks like, if you know what gel electrophoresis is, it kind of looks like this process. So I'm going to talk about that again in a minute. But DNA profiling is helpful when we're talking about the study of forensics. So what's forensics? Give me your crime show definition of forensics.
DNA. It's like scientific analysis for like crime investigation.
Okay. So like sort of using the definition in the book, sort of not. But it is using science to figure out who did what, who done it. Okay, Colonel Mustard in the drawing room with the candlestick. Have you guys played Clue?
No.
Okay. No? I'm sorry, Trinity. Clue is a fun game. I enjoyed that game. Still enjoy it if somebody will play. But the whole way that DNA profiling works is that we can collect DNA from hair, from blood, from skin cells, from other bodily fluids, and then that DNA can be copied a bunch of times and amplified, and then we can compare that to people who are suspected of being the criminal and figure out if yes they are or no they aren't. So I said last week that we can use it to prove innocence or prove guilt. So it does go both ways, okay? Something that DNA profiling is good for then is helping clear people up. So we're going to talk about a case where that happened in a few modules. But part of the problem with DNA profiling is that there are tons and tons and tons of cases where the DNA is just being stored, because even though the technology is much cheaper than it used to be, it still takes time to run the DNA and process it, and it still takes money, and someone's got to pay for it. So that's a potential reason why there's still a ton of DNA sitting in storage waiting for somebody to look at. Okay. So PCR stands for polymerase chain reaction. So tell me the two types of polymerase that we've talked about before. DNA and RNA. Okay. So which kind would be used in a PCR? DNA polymerase, because we're talking about copying DNA. So polymerase chain reactions, and I talked about this last week a little bit as well, take a specific section of DNA that's been cut, so we call that the target sequence, and then when you heat it, you break the hydrogen bonds between the two strands so they separate, and as it cools back down, you put primers in, or have primers in the mixture, so it'll help put down the first sequence of nucleotides. Remember, when you copy DNA, you need RNA primer put down first. So these primers help start that. And then the DNA polymerase that's in the mixture can finish copying the strands of DNA. So we go from one copy that's double-stranded to two copies that are double-stranded. And then we repeat it, and then we have 4. Then we do it again, and we have 8. Then we do it again, and we have
16.
Okay, good. You're with me. Okay. So that's how PCR works. One of the key points that you should know about PCR is that the reason this works is that the DNA polymerase is not actually the DNA polymerase that's found in your cells. Because if we heated the DNA polymerase that's in your body, it would denature because it's a protein. And proteins tend to break apart and the structure disintegrates when you heat them up, which is why cooked meat tastes different than raw meat. Okay? So because the DNA polymerase comes from a bacteria, this bacteria was found in a really hot conditions, like in hot springs. So its DNA polymerase is different, the structure is different. So if we heat it up, it doesn't break down, it doesn't, the technical word is denature. It doesn't unfold. Okay, so that's an important difference between the DNA polymerase in your body versus the DNA polymerase that gets used in this cycle. So you should be able to walk through, here's what a cycle is, and here are the steps to make that work. Okay, but the end goal is to get a bunch of copies of DNA. Okay, that way you have multiple sections of the same sequence. So, gel electrophoresis. This, okay, looks a lot like this. And what was that noise for, Trinity? Bleh. You don't like it. Why? Bad experience?
No, I just don't like it.
You just don't like it.
It's too precise.
There's a lot of precision required. So something that we don't tend to see when we watch crime shows like NCIS and Abby Shudo in NCIS is the precision that it takes. Like we see them talk about results. We don't see how long it takes to run the test, or what happens if they have to like redo it, or what happens if they don't have enough DNA from like the original crime scene to then run, because that can happen. It doesn't take a lot of DNA, but you still have to have a certain amount. What? Yeah, then they just say there's not enough DNA, I guess. Okay. And they try to find something else. Okay. So the way that this works, the gel looks almost like a
Jello.
yeah, kind of like Jello, I guess. And people actually, if you're going to practice for gel electrophoresis, will make a gel out of Jello and then practice injecting into it, because the gel has little wells where you have to use a micropipette. So think like the pipettes that we've used in class, but even smaller than that. So it will measure out a certain controlled amount, and you have to inject that into the well. Then you stick that, yeah, you do have to do it very carefully. If you don't inject it in the right way, or if it leaks, or if you inject it just into the liquid that it's in, like lots of problems come up, okay? But with gel electrophoresis, you put your amplified DNA from the PCR into the wells, and then you stick that gel into a box that you can hook up to a power source. So whether that's a battery or whether you actually plug it in, doesn't matter. But the way you orient or set that up is important, because you want the side the DNA starts on to be the negative side, and the side that doesn't have any DNA in it or doesn't have the wells on it to be positive. And that's because DNA has lots of phosphate groups. And if you remember anything from chemistry or when we talked about the structure of DNA, phosphate's negatively charged. So if you hook this up to a power source, and then you run electricity through the water, this is the electrophoresis portion of it, it's using electricity to power this, it causes the DNA to move down the gel, because negative would be attracted to positive. So the DNA strands move because of the phosphate groups. The bigger the strand is, or the longer the section of DNA, the slower it's going to move. So that means that when over time, an hour, maybe 2 hours, as you let the gel just run, you get bands separating out the different sizes of DNA, or the different lengths of DNA. So the lengths that are longer are closer to the well. The lengths that are shorter end up at the end of the well. It's also possible to have this process happen and have DNA run off the end, because you can run it so long that the DNA just like comes out the other side of the gel. So that's a possible problem with electrophoresis. You do have to watch it and make sure you're not running it too long, okay? But based on that, we would call each of these lanes, we can look at specific bands and compare them to, for example, this, and look at, okay, we have this DNA from the crime scene, there's this suspect and this suspect, these bands don't match this. So this suspect isn't the one who done it. But suspect 2, they match. This is the guy who done it. Or at least that person was there, or their DNA was left behind. Whether they did it or not might be different, depends on where the DNA is found, okay? So that's how gel electrophoresis is used. Questions on this? Okay, so let me ask you, why do large molecules move more slowly than the small ones? Because what? Okay, they are bigger. Why does that mean they move more slowly? Okay, yeah, that's true. If you're gonna push 50 pounds down the hallway versus 10 pounds down the hallway, you'll push the 10 pounds more quickly. Okay, and that's pretty much it. A more technical answer would talk about the charge in the phosphates compared to like moving through the gel, but it's correct. The bigger it is, the harder it is to push through the gel. Okay, cool. Next section. Okay, when we use gel electrophoresis, we aren't sequencing the entire genome. So this is not doing what the Human Genome Project did and taking all of the nucleotides, all 6 billion of them, and figuring out the order. Okay, this is using STRs, or short tandem repeats. Okay, so this is using just specific sections of DNA, copying those or amplifying those a bunch, and then running those in gel electrophoresis. Okay, so these STRs, or short tandem repeats, are different in different people. Do you remember how much of your DNA is actually expressed? Less than 10%? Really small amount. So the rest of the DNA in your body is something that a lot of researchers will call junk DNA. And we're learning that's not really junk, and it does actually have a purpose a lot of the time. We just haven't figured out what it is yet. But it's not DNA that is used to make proteins. So in that junk DNA and that extra stuff, you have short tandem repeat fragments or sequences. So in your genome, you might have a short tandem repeat on chromosome 1 in location 1 that has 12 sequences, and mine might have 20. And somebody in 6th period might have that sequence that only has 6 repeats. So it's the idea that you have sequences of DNA or pieces of your DNA that repeat different lengths of time or different amounts of time compared to other people. And if we know where these STRs are, we can compare several different STRs in a gel electrophoresis sequence to figure out who is, what your DNA looks like. So the longer your STR fragment, the heavier it would be, so then the closer to the well it would be. Make sense? What's STR stand for? Short tandem repeat, okay. So STR analysis, again, is using that to profile DNA. Okay. I think that's pretty good. I don't need to elaborate on that anymore. So then this guy, I said we were going to talk about a guy who was exonerated. That would mean like his name was cleared. This was a guy who was on death row, if I'm remembering correctly, and they did short tandem repeat analysis on his DNA compared to the DNA found at the crime scene, and found out he wasn't the guy. Okay, so he was tried, he went through the whole trial, found guilty, in jail.
What if they would have killed him?
Found out that then he would have been killed wrongly, and that does happen. So there's a whole ethical debate for, do we have death row, do we have death sentences or not? That's one argument for not, because people can be convicted wrongly.
Does it happen?
It does happen. I guess that means, depending on what you mean by a lot, I'm quoting from here, okay? Lawyers at Innocence Project, which is a nonprofit that's dedicated to using DNA to try to prove people's innocence and exonerate them, exonerated more than 340 convicted criminals since 1989. Okay, so that's like my lifetime, not quite your lifetime. But 17 of those people were on death row. So does it happen a lot? That's in different states. I don't know how many people were on death row in all of those states. Some states have the death penalty, some don't. Okay, so does it happen? Yes. Does it happen to everyone? No. But yeah, it's not right to have people killed when they didn't do it. So that's one argument people make for not having the death penalty. We also use DNA profiling for more than just like NCIS criminal work. DNA profiling is what makes a paternity test possible. So you can figure out that someone is such and such's baby because of DNA profiling. There are other ways you can do that. So remember, we've talked about blood typing before. People have figured out that they're adopted through blood typing. So it doesn't have to be DNA profiling, but that would be evidence that's admissible in court. So the example that's given here is a guy who, I don't remember what it was. I think he was not Cheddar Man and not Thomas Jefferson, although both of those are mentioned here. Yeah, his name's, the Cheddar Man was from Cheddar, England, so he's called the Cheddar Man. But he was a skeleton found in England, and they took DNA from the bone marrow of the skeleton, ran it, profiled it, and then found that he had a descendant, like, living 60 miles away from Cheddar, England. So his family apparently stayed in the area, or at least some of it did. But we've also used things like this to prove once someone who's famous or well-known dies, and somebody says, this kid is, like, this person was my baby daddy. Proving it that way. Yes, I saw that face. Yes. Okay, there you go. He just thought it was weird that I used that expression. And, you know, I gotta keep it interesting for you, make you laugh. But this is also how we proved that Thomas Jefferson did have personal relations with some of his slaves, because they did paternity testing. So you can figure things out many, many years after the fact using DNA profiling. Okay, so that's one example. We can also use DNA profiling for more than just humans. So you can do it for animals or potentially plants. I don't know why you would do it for plants, but I guess you could. Maybe you would do it to do some of the stuff that Abby Shudo does and figure out like this person's drug is actually the same as like the drug from the crime scene. I don't know. Maybe that's when we use DNA analysis on plants. But their example here is putting poachers in jail for poaching tigers because they found flesh from the tigers under the poachers' nails. So they were able to link it to, we know these tigers were killed. They had the DNA under their fingers and they did it. Okay. Yeah, that is, again, poaching is illegal. You shouldn't do it. Okay. So genomics. Do you remember from the video we watched last week, that little USB drive that they started the video out with? This is not a normal USB. It's from a company called Nomi. No? Some of you were like, yeah, some of you are like, nope. Okay, that USB drive is an example of a third generation sequencing technique. So first generation sequencing would be like the Human Genome Project. It took 13 years and like $100 billion to sequence the whole human genome first time around. Okay, second generation sequencing techniques would look at taking machines like this and sequencing all of the nucleotides in a row. So this is not short tandem repeat analysis, just pieces of DNA. This is looking at the entire genome for one human. Okay. Third generation techniques take the whole strand of DNA and thread it through a little tiny hole. So like a nanopore size, really, really small. And as they sequence that, they can figure out what the entire sequence is in order. So every time we develop new technologies, we can make the sequencing cheaper and faster and then easier. So eventually it might be possible to sequence everybody's entire genome. Right now, if you paid for the test from 23andMe or Ancestry.com or one of the other companies like that, they would not be looking at all of your DNA. They would only look at like 1 million different sites in your chromosome. Just 1 million, which is a whole lot less than all 6 billion nucleotides. So I say just 1 million, it really is a lot, but it's not as much as all of your DNA is, okay? So you should kind of know the difference between those different generations of sequencing techniques and how they work. And if you have a question about that, we can talk about that in a minute. Again, we can come back to it. Genomics then specifically, because this chapter is about DNA technology, biotech, and genomics. Genomics is studying the entire set of the genome. So this is only possible if you can sequence the entire genome, okay? The reason that a lot of scientists are interested in trying to do this is looking at how related different organisms are to each other. So if we look at humans compared to chimpanzees, how many differences are there in our DNA sequences? Or if we look at humans compared to whales, how many different nucleotides are there in our sequences? So when you're looking at this graph, tell me what it's measuring.
The number of genes.
Yeah, number of genes. So this is in thousands, okay? Compared to, what's this?
The
Size of haploid genomes. So haploid versus diploid. Do you remember what that is?
2 versus 1.
Usually. So our cells are normally diploid. So that number 23andMe comes from the haploid number of our chromosomes. We have 46 chromosomes, but the haploid number, the number you get from each parent, is 23. So the haploid genome is how many billions of base pairs are there? Humans are up at like 3.5 billion in just half of your DNA. So then the other half would bring us up to like 7 billion. Okay, just because you have a bigger genome though, just because you have more chromosomes or longer DNA sequences, doesn't mean that you have more genes. Okay. What's a water flea? Type, oh, sorry, that's my phone. Normally I turn it off. Okay, there we go. I don't know, some random number. It's great. Okay, this is, yes, firsthand like experience, don't leave your phone on because it will ring when you don't expect it to. So humans actually don't have that many genes. Okay, total you have like 21,000 genes. And that's your diploid number of genes, not your haploid number of genes. What organism on here has the most genes?
Rice.
Yeah, rice has more genes than we do. Okay, so having a longer DNA sequence, having a bigger genome, does not correlate with having more genes.
And the mustard.
Okay, yeah, the mustard plant has more. A lot of plants actually have more genes than we do. A water flea is a type of insect. And I can look up a picture when I'm done if you want.
Please don't.
You're good? You don't want to see it? Okay, Human Genome Project. Do you remember how much I said this cost?
35,000.
Oh no, it was way more than thousands of dollars.
Millions.
Billions. Billions. I believe that it is in billion. Like,
Don't say designer baby.
yeah, it was mentioned on the Designer Babies episode that we watched. It was mentioned in the video we just watched last week. Okay, but it took 13 years to sequence the first human genome. Okay, and what we're finding, remember I mentioned junk DNA. Okay, of all of the DNA that you have, very little of it actually codes for the proteins that your body makes. So this little section here, the 1.5%, that's what's going to make protein. So the introns, those would be transcribed, but remember they're cut out. So that's a good chunk. So total between the regulatory sequences and the introns and exons, we've got like 25%. But not all of that ends up being protein. Only like 1.5% is protein. So we've got repetitive DNA. This would include the STRs, the short tandem repeats. Okay. We've got other types of repetitive DNA, and then DNA that doesn't code for anything, that's there for some other reason. And scientists are still figuring out what all of those reasons are. So what does all of this other DNA do? We want to say that it's junk DNA, but we know it's really not junk, because if you have mutations in those areas or you're missing those areas, then there are problems that show up. So it's not truly junk DNA. We just don't know how it all works together yet.
Is that the 15%?
Okay. Is that the what?
Is that the 15% or 44%?
The 15%.
Which percentage?
Yeah.
The 1.5%.
Oh.
Not 15%. Did I say 15?
I thought you pointed to the blue one.
Sorry.
I was just wondering.
Go. So you're wondering which part is the junk DNA? The some people might call introns junk. The repetitive stuff could potentially be called junk. But we're figuring out that although it looks like it's all junk, it's really not all junk, because there's some reason for it. If you don't have it, like a normal human, then you don't develop the right way. So it's not really junk, even though we think it might be junk. Okay. So yeah, other information from here talks a little bit about genetics. So it talks about haploid versus diploid. Your haploid chromosomes, we have the sex chromosomes, which are the last pair, so number 23. Females would be XX, males would be XY, but all of the other chromosomes are called autosomes. So there's the sex chromosomes, and then there's the autosomes. So you have 44 autosomes, or 22 pairs of autosomes, and you have one pair of sex chromosomes. Okay, that's a typical haploid or diploid number, so you should know that. Moving on, then we get to talk about the whole genome shotgun method, which is taking an entire chromosome and you chop it up into little tiny pieces. Remember, we talked about restriction enzymes last week. So those are proteins that will cut the DNA at specific sites. So you get from one chromosome a specific number of fragments, and then you can sequence those fragments. So you put all of those pieces into a test tube and allow them to get lined up again, and you reassemble the whole thing. So this is a different way to sequence the whole genome, and that's something that's becoming much more affordable. So it took 13 years, it cost $100 million. Now we can do this for less than $1,000. Huge difference. Doesn't mean people want to pay $1,000 to have this done, but remember the 5-year-old from the video last week. They were trying to figure out what was wrong with him. Cost less than $1,000 to sequence his whole genome. Okay, so for cases like him, that's when we use this. That's why it becomes important. Okay, Another new term, bioinformatics. This is pretty much you have all of this data. Now, what do you do with it? So this is data analysis, but this is data analysis on a level where you're not doing it by hand. So you're not taking a graph, or a chart and just analyzing it on your own. You're using an Excel spreadsheet or a program to analyze it. Okay, so that would be bioinformatics. You guys are going to get to practice doing stuff like this with a virtual lab later this week. Okay. Hi, how you guys doing?
Hi, good, how are you?
Great. The last section is talking about how genomes can be used to help trace evolutionary relationships. So this is what I mentioned. A lot of biologists are interested in this because it allows us to see how closely related different things are. So if we sequence a human's genome, we can also sequence a chimpanzee's genome and see how many changes there are. And that allows us to see if we're more closely related to a chimpanzee versus a Neanderthal, which would be a different type of organism in the Homo genus. Okay. So, do you have questions that you want to ask while there are other people watching?
No pressure.
Yeah, no pressure.
With us in here, they're probably not going to answer.
Yeah, probably not. So,
Trinity,
you know.
why don't you ask a question?
I don't have a question.
You sure?
Yeah.
Thank you.
thanks, guys.
Thank you so much.
Yep. Huh? They're here for STEM. So do you guys know what STEM stands for? Some of you, yes. Science, technology, engineering, math. They're here to help us see what qualifications we meet to be a STEM certified school. To have just random people come in. The door was open. They didn't even have to knock. Okay. So now do you have any questions? No? Okay. I will stop